Hello, I'm Roger Hallman, a doctoral student at Dartmouth College and a computer scientist with the Naval Information Warfare Center Pacific. My co-authors, Anjali, Chikkula, Prioleau, and I thank you for attending the presentation of our paper, Predicting Criticality in COVID-19 Patients. The agenda for this presentation includes introductory and background material on both COVID-19 and prognostic models, then a discussion of the data and methodology that we used to build our own prognostic model, and finally, we provide results and a brief discussion of our model's strengths and limitations. I'm sure that I don't need to cover much background information on COVID-19. As of the time of this recording, there have been more than 22 million COVID-19 cases worldwide, with nearly 15 million recoveries and 780,000 recorded fatalities. The preferred COVID-19 diagnostic method is the reverse transcriptase polymerase chain reaction test. CT scans results have also seen success in COVID-19 prognosis, although it is worth noting that the American College of Radiology recommends against using CT scans for diagnosis. Proxy diagnosis models are useful where other methods aren't available. None of these methods are flawless. Reverse transcriptase tests have shown significant false negative and false positive rates, thus best practices may include consistent results over a series of 3 tests. There's currently no approved vaccine nor recommended antiviral therapeutic for COVID-19, though progress is being made. Barring widely available vaccines, studies predict multiple waves over the next couple of years. Therefore, we believe that clinical management will remain crucial for COVID-19 care. We are aware of at least 31 predictive models that have been developed and described in technical literature since the emergence of the pandemic, most of which use data from Chinese hospitals. These models, which utilize both clinical markers as well as CT scan features, may predict mortality risk or hospital admittance. There are also a handful of models which rely exclusively on clinical markers and data other than CT scans for admittance and prognosis predictions. Most models lack transparency and are known to be at high risk for bias due to non-representative samples and exclusion of other patients. Our paper comes out of a course on the healthcare applications of data science that Temi teaches at Dartmouth College. We read several papers on COVID-19 prognosis prediction during the class and sought to answer a very standard basic science question. Are these results repeatable? To that end, we sought to build a COVID-19 prognosis predictor that can assist clinicians with patient routing that verifies the repeatability of earlier samples and strives to adhere to the best practices of model transparency. All of our code and data are publicly available in a Git repository. The link is in our paper. There is a paucity of available COVID-19 clinical data. We use a data set from the Hospital Israelita Albert Einstein in Sao Paulo, Brazil, that contains information from 5,644 patients seen between March 28th and April 3rd. Of the patients seen, 558 tested positive for COVID-19. The data set is anonymized with patients denoted by an ID number, as well as one of 4 admissions classes. 112 clinical features are provided, all of which have been standardized with a mean of 0 and a standard deviation of 1. No raw descriptive statistics are provided. This data set has 4 admission levels, which we use for our criticality measure. Level 0, the patient is not admitted to the hospital and is instructed to quarantine at home. Level 1, the patient is admitted to the hospital's regular ward for monitoring. Level 2, the patient is admitted to a semi-intensive care unit for monitoring and a variety of treatments, for instance, non-mechanical ventilation. Level 3. The patient is admitted to an intensive care unit for emergency intervention. Unfortunately, this data set turned out to be rife with missing and imbalanced data. Only 6 features were complete, and no patient has a complete set of features. This is mainly due to the fact that there were not standard clinical protocols and different doctors ordered different tests. About 90% of COVID-19 patients in our dataset were directed to quarantine and convalesce at home, while only 2.8% were admitted to either the semi-intensive care unit or the intensive care unit. To deal with missing data, we first tried to find features strongly correlated with criticality using imputed data. This showed only criticality strongly correlated with itself, so using imputed data is not an ideal way to proceed. On the other hand, we found 9 strongly correlated features using unimputed data. Thus, we need a model that is adept at handling missing data. Our correlation heat map is on the next slide. Here we see the features that have the strongest relationship with criticality. We use XGBoost, a tree boosting algorithm that is efficient, flexible, and portable. Moreover, XGBoost is adept at handling missing data and constructing an explainable decision tree model. We were partially inspired to use XGBoost through reading literature where it was used in Yan et al.'s clear and explainable model, as well as the success that XGBoost has seen in various Kaggle competitions. Our methodology is shown on the next slide. This slide illustrates our entire methodology. We split the data into training and test sets and do 10-fold cross-validation as you would expect in a class. We use XGBoost's feature importance algorithm and construct our model with the 5 most important features. This confirms some of the repeatability of earlier works, specifically C-reactive proteins, lactic dehydrogenase, and alanine transaminase are important features in multiple prognosis models. Our model, constructed on standardized data, gives criteria for each prognosis class. For patients not admitted, it seems fairly obvious that inflammatory markers should be low and antibody counts should be around normal, neither too high nor too low. For patients admitted to the intensive care unit, inflammatory markers are severely elevated and other readings are way off as well. The middle 2 classes and their selection criteria may be a little bit more subjective, partially due to the fact that semi-intensive care units may not be used everywhere. Since this model is constructed on standardized data and no descriptive statistics have been provided, this model is not immediately useful outside Sao Paulo. Our model is only as good as the data that it was trained on, and here we see the effects of our initial data imbalance. Our model is very good at predicting which patients will be sent home, but its performance is much more variable when it comes to other prognosis classes. We believe that this can be resolved with more data to train on. In an effort to make our model more useful to the broader community, we conducted a literature review to aggregate descriptive statistics for our features over a broader population. C-reactive protein and other inflammatory markers turn up repeatedly, so there are many populations to aggregate from. On the other hand, the blood acid base ratio is a bit more obscure, and I only found a handful of reports on COVID-19 treatment that recorded this information. Here we see our model incorporating the aggregated descriptive statistics from the previous slide. Though we strove to create a prognosis prediction model that would be useful to assist clinicians, we've covered a number of limitations and challenges that must be overcome before it will become in any way effective or ready for use. There is generally a lack of publicly available raw data for COVID-19 patients and treatment. Moreover, the data that is available is incomplete and imbalanced. In our case, the data set was also standardized with no descriptive statistics for any features. Nevertheless, we believe that our general approach is valid and worth pursuing further, for instance, by retraining our model with more data. I would like to give a quick acknowledgement to the DoD Smart Scholarship, which funds my studies at Dartmouth College. Also, I would like to give a quick shout out to Professor Temiloluwa, who is leading an effort to understand recovered COVID-19 patients' experiences, especially patients who convalesced and recovered at home. You can learn more at www.c19insider.com. Thank you for your attention. I am happy to answer questions. If you are watching this presentation after its conference slot, then feel free to email me at the email address presented, and I will try to get back to you in a timely manner.